There is a possibility, small but real, that the body you live in has never belonged entirely to you. Not in a metaphorical sense, not in the philosophical sense of wondering who you really are, but in the most literal biological sense possible. The cells in your body, the ones that carry your DNA, the ones that make you genetically you, may not all be carrying the same instructions. Some of them may be running a completely different genetic program. Some of them may belong, in every measurable biological sense, to someone else. That someone else never lived outside the womb. They were your twin, and they vanished before you were born, absorbed into you so completely that no memory of them exists anywhere except inside your own cells. This is not science fiction. This is a documented biological phenomenon called human chimerism, and it is stranger, more common, and more consequential than almost anyone outside of genetics research knows. Welcome to Sleepy Science Stories. Tonight we are going inside the body, not at the scale of organs or tissues, but at the scale of cells and DNA, the level where identity is written in a chemical alphabet, and where the boundary between one person and another turns out to be far less clear than we assume. The word chimera comes from Greek mythology, where the chimera was a monstrous creature with the head of a lion, the body of a goat, and the tail of a serpent, a single being assembled from the parts of different animals. In biology, a chimera is an organism that contains two or more genetically distinct populations of cells, all derived from different original sources, all living together inside a single body. In humans, this happens in several ways, and for several reasons, and with consequences ranging from invisible to life-altering. Some chimeras never know what they are, and live their entire lives carrying a second genome in their cells without the slightest indication that anything unusual has occurred. Others discover their condition only in the most dramatic circumstances possible, when a DNA test tells them that they are genetically not the mother of their own children, or when a criminal investigation produces evidence that points to a person who could not possibly have committed the crime, at least not the version of that person whose DNA was sampled. These are not thought experiments. These cases have happened. They have ended up in courtrooms and medical journals and international news coverage, and each one has forced scientists, lawyers, and ethicists to reckon with a question that chimerism makes newly complicated. What does it mean to be one person? Let your breathing slow. Let the weight of the day settle out of your body. Tonight we are going somewhere quiet and strange, into the interior of what it means to be you, and what it would mean if the answer to that question turned out to be more complicated than you ever imagined. The science we will cover tonight sits at the intersection of genetics, immunology, developmental biology, and philosophy. It is not speculative. Every case described in this episode is documented in the scientific and medical literature. And the phenomenon of chimerism has been studied rigorously enough that we now understand its mechanisms with considerable clarity, even if the full scope of its prevalence remains uncertain. What makes chimerism remarkable is not that it is exotic, but that it is hidden, woven into the fabric of ordinary human biology, present in ordinary people living ordinary lives, visible only when circumstances force a closer look at what the cells of the body actually contain. Tonight we will take that closer look together. To understand chimerism, it helps to start with the most basic picture of what a human body is, genetically speaking. Every cell in a typical human body contains a nucleus, and in that nucleus is a copy of the genome, the full set of genetic instructions that defines the biological identity of that organism. For most people, every cell in the body carries essentially the same genome, because every cell descends from the same original cell, the fertilized egg that formed when one sperm combined with one egg. That fertilized egg divided and divided and divided again over 9 months of development, and every division copied the genome forward, so that the trillions of cells in the adult body are all, genetically speaking, copies of that original single cell. In a chimera, this is not the case. A chimera has two distinct populations of cells, each carrying a different genome, descended from two different original sources. One population might make up 60% of the body's cells, and the other 40%, or the split might be 90-10, or 50-50. And the two populations are not randomly mixed throughout the body. They can be, and often are, distributed in distinct patches, with one genome dominating certain tissues and organs, and the other genome dominating others. This means that a chimera is not one person in the genetic sense, but two distinct genetic individuals occupying the same body simultaneously, sharing the same brain, the same heartbeat, the same face, and the same continuous stream of conscious experience, while carrying two different genetic identities distributed across their tissues in a pattern that varies from person to person, and that can only be detected by sampling multiple tissues and comparing the results. The myth of the chimera described an impossible creature stitched together from pieces of different animals. And there is something of that image in the biological reality. Except that human chimeras are not stitched together from pieces. They are blended at the cellular level, a mosaic of two different genetic identities, woven into a single living body so completely that from the outside, and usually from the inside as well, there is no way to tell that anything unusual has happened. The biological mechanisms that produce chimerism are varied, and the consequences of each mechanism are different, but all of them trace back to the same fundamental fact about early development. In the earliest days after conception, the boundary between what will become one person and what will become another is not yet fixed. Cells mix, merge, and communicate in ways that can permanently alter the genetic composition of the developing organism. The window during which this can happen is brief, measured in days, but what happens during that window can persist for an entire lifetime. In mythology, the chimera was a monster. In biology, chimeras are people, living ordinary lives, largely unaware that they carry a biological secret encoded in their own cells. A secret that only the most careful genetic analysis can reveal, and one that challenges some of our most basic assumptions about what it means to be a unique individual. The word individual itself comes from the Latin meaning indivisible, not capable of being divided, a single unified entity. The chimera is, in the most literal sense, a challenge to that word's meaning, because the chimera is, at the cellular level, divisible into two distinct genetic populations, and yet the person who is a chimera is not experienced as divided, does not feel divided, has no awareness that the cells making up their body are following two different sets of instructions. The division exists only at the molecular level, invisible to introspection, readable only by instruments designed to look for it. This combination of biological complexity and experiential simplicity is what makes chimerism so philosophically interesting and so practically important. The biology is real and has real consequences, but the person living that biology has no direct access to it, and the story they tell about themselves, the story of being one continuous self, is not wrong. It is simply incomplete. The most common route to human chimerism begins with twins. Twin pregnancies are more common than many people realize. Depending on the population and the method of conception, between 1 in 30 and 1 in 80 pregnancies begins as a twin pregnancy, meaning two embryos implant in the uterus at the same time. But not all twin pregnancies end in the birth of twins, because one of the most common events in early pregnancy is the spontaneous loss of one of the embryos, usually in the first trimester, often before the mother even knows she is carrying two. When this happens, the surviving twin absorbs the cellular material of the vanished twin, and this absorption is not merely the recycling of organic matter. It can include living cells, cells that carry the vanished twin's genome, and those cells can integrate into the surviving twin's developing tissues, taking up residence and continuing to divide and replicate alongside the host twin's own cells. The result is a person who carries two genomes, their own and a remnant of the twin who never survived to be born. This phenomenon is called the vanished twin syndrome, or more precisely, the surviving twin of a vanished twin may become a chimera. Not every case of vanished twin produces chimerism. The timing matters, and the nature of the absorption matters, and the fraction of cells that successfully integrate into the host's tissues varies enormously. In some cases, the absorbed cells are simply metabolized and leave no trace. In others, a significant population of the vanished twin cells takes up permanent residence and persists throughout the host's entire lifetime. The vanished twin is not entirely gone. Something of them remains, scattered through the organs and tissues of the sibling who survived, encoded in DNA, but not in memory, present in the body but absent from the story that body tells about itself. There is something quietly extraordinary about this. The person who was once two is now one, but the one carries the other inside them, not as a ghost in any mystical sense, but as a biological reality. A second set of genetic instructions expressed in some fraction of their cells. The vanished twin's DNA is inside the liver, or the skin, or the bone marrow of the surviving sibling, doing the ordinary work of keeping cells alive, completely integrated into a body it never expected to inhabit. The prevalence of the vanished twin syndrome is difficult to measure precisely because it requires early ultrasound confirmation of a twin pregnancy, followed by the later loss of one embryo, and the chimerism it might produce is essentially invisible without deliberate genetic testing of multiple tissues. But estimates from studies that track twin pregnancies from the earliest weeks suggest that the vanished twin may be responsible for a significant fraction of all human chimerism, and given the frequency with which twin pregnancies resolve into singleton births, the number of living chimeras who carry a remnant of a lost twin may be substantially higher than most people imagine. There is something in this that resists easy categorization. The surviving twin did not lose a sibling in any conventional sense, because the vanished twin never drew breath, never had a name, never existed outside the womb in any form the surviving person could remember or grieve. And yet the biological trace of that early companionship persists, encoded in cells, present and functional, carrying the vanished twin's DNA in tissues that may span the entire body. The grief, if it can be called that, is cellular rather than conscious, biological rather than experiential. A memory written not in the mind, but in the body itself. The vanished twin is not the only route to chimerism, and understanding the full range of mechanisms helps clarify why chimerism is more diverse and more widespread than a simple twin loss story suggests. The most fundamental form of human chimerism is called tetragametic chimerism, meaning it arises from 4 gametes, 2 eggs and 2 sperm, rather than the usual 2. In a typical pregnancy, 1 egg is fertilized by 1 sperm, producing 1 zygote that develops into 1 person. In tetragametic chimerism, 2 eggs are fertilized by 2 different sperm, producing 2 separate zygotes, which then fuse at an extremely early stage of development, before they have had time to become distinct embryos, and the result is a single organism built from 2 genetically distinct cell lines. This is what the mythological chimera actually represents in biology. A single body assembled from 2 separate originals. The key difference from the vanished twin scenario is that in tetragametic chimerism, neither cell line originated from the other. They are 2 completely independent fertilization events that happened to merge before the developmental process had established them as separate individuals. The fusion must happen very early, within the first few days after fertilization, when the embryo is still a small ball of cells with no fixed fate. At this stage, cells are still largely interchangeable, their ultimate destiny undetermined. An emerged embryo can develop normally because the 2 cell lines are mixed so early that the developmental process simply treats the combined population as a single organism. The resulting person has no symptoms, no malformations, no sign of anything unusual. because the developmental program that builds a human body from a fertilized egg is robust enough to work with a heterogeneous cell population, provided that population is present from the very beginning. A third mechanism is maternal-fetal microchimerism, which we will explore in depth later, where cells pass between a mother and her fetus during pregnancy, in both directions, and persist in the host for years or even decades afterward. A fourth mechanism is chimerism acquired through organ transplantation or blood transfusion, where donor cells take up residence in the recipient's body and survive long after the transfusion or transplant. Each mechanism produces a slightly different pattern of chimerism, with different tissue distributions and different genetic signatures, but all produce the same fundamental result: a single body containing cells from more than one genetic source. What all these mechanisms share is an exploitation of the early embryo's essential flexibility. In the very beginning of development, cells are not yet committed to a specific fate. The boundary between self and other has not yet been established, and the immune system that would normally attack foreign cells has not yet learned to distinguish self from non-self. The window is brief, but during it, the rules that govern individual genetic identity are temporarily suspended, and two can become one. The fact that this window exists, and that it is exploited by multiple different biological mechanisms, suggests that the rigidity of genetic identity is a developmental achievement rather than a given. A body does not begin with clear boundaries and a single genome. It begins with a cluster of cells with no boundaries at all, and the process of development progressively establishes those boundaries, committing cells to fates, calibrating the immune system to recognize self, drawing the line between what is inside and what is outside. Chimerism is what happens when something crosses that line before it has been fully drawn, and persists after it has been. The existence of multiple distinct routes to chimerism, all exploiting the same early developmental window, suggests that this kind of boundary crossing is not a rare accident, but a recurring feature of early human development, one that leaves traces in a significant fraction of the population, and that has shaped the biology of our species in ways that researchers are only beginning to understand. One of the places chimerism most often reveals itself, when it reveals itself at all, is in the blood. Blood type is determined by a small set of genes that instruct blood cells to display certain proteins on their surface. The most familiar system is the ABO system, which divides humans into 4 blood types based on which combination of proteins their red blood cells carry. Your blood type is fixed at conception, encoded in your genome, and every red blood cell your bone marrow produces should display the same blood type, because every cell in your bone marrow carries the same genome. In a chimera whose second cell line has taken up residence in the bone marrow, this is not what happens. If the chimera's own genome codes for blood type A, but the absorbed twin's genome codes for blood type B, the bone marrow will produce a mixture of blood type A cells and blood type B cells, and a blood test will return an anomalous result, showing a person with both blood types simultaneously, which is not supposed to be possible. This is how many chimeras are first discovered. A routine blood test returns an unexpected result. A person is shown to have 2 blood types, and the investigation that follows eventually reveals the genetic explanation. The person is not sick. Nothing has gone wrong. Their blood is simply a mixture of 2 populations of cells, each faithfully following the instructions of a different genome. The first confirmed human chimera on record was discovered in exactly this way. In 1953, a British woman donated blood and was found to have a mixture of 2 different blood types, which was so unusual that her doctors suspected a laboratory error. When the test was repeated and confirmed, an investigation eventually determined that she was a chimera, carrying 2 cell lines in her blood, one her own and one belonging to her twin brother, who had died shortly after birth. Cells from her brother had crossed the placental barrier during their time together in the womb, taking up residence in her bone marrow, where they had continued producing blood cells of his type for the entire duration of her life. She was 61 years old when this was discovered. Her brother had been dead for 60 years, and yet his cells were still alive inside her, still making blood, still expressing his genetic identity in a body that had outlived him by 6 decades. Blood type is perhaps the most concrete demonstration of what chimerism actually means at the cellular level. It is not an abstract genetic difference, but a measurable functional difference in the cells themselves. 2 populations of blood cells, carrying 2 different surface proteins, flowing together through the same circulatory system, doing the same job while following different genetic instructions. A body that is, in this specific measurable way, 2 people at once. The British woman discovered in 1953 was not the last person to be found this way. As blood banking and tissue typing became more sophisticated through the latter half of the 20th century, additional cases of blood type chimerism were identified, and each one prompted the same cascade of investigation and the same eventual conclusion. The anomaly was not an error. The person was simply 2 in the only sense that matters at the cellular level, and that duality had been present from before they were born and would persist until the last of those borrowed cells completed the span of their lives. Most people with chimeric blood types never cause confusion, because the mixed blood type, while anomalous, is not medically dangerous in the way that an incompatible transfusion would be. It is a biological curiosity rather than a clinical problem, and for many chimeras it remains permanently undiscovered, their unusual blood quietly circulating through a body that has never been asked to explain it. Of all the cases in which chimerism has surfaced in human lives, none is more disorienting than the case of a mother told that she is not genetically related to her own children. This has happened. It has happened more than once. And the story of how it happens, and what it means when it does, is one of the most striking illustrations of why chimerism matters beyond the laboratory. The most widely reported case is that of Karen Keegan, a Boston woman who in the late 1990s needed a kidney transplant and had her children tested as potential donors. Tissue typing, which assesses genetic compatibility for transplantation, showed that Karen's children were not a genetic match for Karen. Not in the way a child is sometimes an imperfect match for a parent, but in the more fundamental sense that the genetic markers used for tissue typing were not shared between Karen and the children she had carried and given birth to. The initial assumption was that there had been a laboratory error. The tests were repeated. The error was not found. A more extensive genetic investigation was launched, and the results became more puzzling rather than less. Different tissues from Karen's body produced different genetic results. Her blood had one genome. Her thyroid cells had a different genome. Her skin cells and her bladder cells and cells from other tissues each produced slightly different results, some pointing toward one genetic identity and some toward another. Karen was a chimera. Her body contained 2 distinct cell lines, and the second cell line, not the one present in her blood, was the line that had produced her eggs, which is why her children were genetically related to that second cell line and not to the genome found in her blood. The maternal relationship was real in every biological sense. The children had grown inside her body, had been nourished by her blood, had been born from her. But a standard DNA test, sampling only her blood, would conclude that she was not their mother. The implications are vertiginous. The test was not wrong. The conclusion was wrong because the test was incomplete, because it sampled only one tissue from a body that contained 2 distinct genetic identities. And the tissue it sampled happened to carry the genome that was not the one that mattered for reproduction. Karen's case was eventually resolved, and her maternity was legally established, but the case raised questions that have not been fully answered. How often does this happen, undetected? How many maternity or paternity disputes have been resolved incorrectly because DNA testing sampled the wrong tissue in a chimeric individual? How many legal determinations of biological identity rest on genetic evidence that may be accurate for one part of a chimeric person's body, but not another? The case of the mother who wasn't is also a case of science running into the limits of its own assumptions. Assuming that a person has one genome, that sampling any tissue will give you that genome, and that the genome you find will be the one that determines biological relationships. Chimerism breaks all three of those assumptions simultaneously. Karen was not unique in her experience. Other women have been told by DNA testing that they are not the biological mothers of their children, and in each case the same pattern of investigation eventually revealed the same biological explanation. The frequency of these discoveries is difficult to estimate, because most would only surface if DNA testing were performed for some reason, and in most pregnancies no such testing occurs. The cases we know about are those where an anomalous result was pursued rather than dismissed, and they are almost certainly a small sample of the total. The legal system's encounter with chimerism is not limited to questions of maternity and paternity. It has also surfaced in criminal investigations, where the fundamental premise of DNA forensics, that a person has one unique genetic identity, and that any sample of their DNA will prove who they are, has collided with a biological reality that the legal system was not designed to accommodate. The case that first brought this to wide public attention involved a man in the United States who was investigated in connection with a sexual assault. DNA evidence from the crime scene was tested against a database of known offenders, and the search returned a match. The man was identified, interviewed, and his DNA was sampled for confirmation. The confirmation test failed. His DNA did not match the crime scene evidence. Further investigation revealed that the match in the database had come from a previous blood sample, and the new sample had come from a cheek swab. And the two samples did not match each other. The man was a chimera. His blood and his cheek cells carried different genomes, and the crime scene sample matched neither of the two genomes found in his body, or matched one but not the other, producing a result that standard forensic procedure was not equipped to interpret. Another case involved a woman named Lydia Fairchild, who applied for public assistance in Washington state and was required to provide DNA evidence of her relationship to her children as part of the application process. The DNA test showed she was not genetically related to children she had clearly given birth to, because there were witnesses, including the obstetrician who delivered them. The state initially suspected fraud, believing she might be acting as a surrogate or had somehow falsified the birth records. A court-ordered observer was present at the birth of her next child, confirming she was the biological mother, and yet the DNA test on her blood still showed no match to the child. Lydia was also a chimera, and the cell line in her blood was different from the cell line that had produced her eggs and her children. And the case took years to resolve through exhaustive testing of multiple tissues across her body before her biological maternity was legally confirmed. What these cases illustrate is that the forensic and legal systems built around DNA evidence rest on an assumption that chimerism renders potentially unreliable. The assumption is that every person has one genome, that any tissue will reveal it, and that the genome is the person. For chimeras, none of these things is fully true, and the legal system has been slow to develop protocols for handling cases where the genetic evidence is internally inconsistent because the person being tested has two of them. The Lydia Fairchild case attracted significant media attention and was later featured in a documentary, and the coverage helped raise awareness among both the public and the legal profession that DNA evidence, while extraordinarily powerful, is not infallible in the way it had sometimes been treated. The case did not undermine confidence in DNA forensics generally, because the anomaly is rare and the circumstances that produce it are specific, but it demonstrated that the interpretation of genetic evidence requires understanding the biological complexity of the individual being tested, and that complexity is not always what we assume it to be. Each of these cases also reveals something about the assumptions embedded in the systems that govern our lives. Legal systems, medical systems, forensic systems, all rest on a model of the individual as genetically singular and as representable by any tissue sample taken from them. Chimerism is the exception that tests that rule, and in testing it, reveals that the rule was always a simplification that happened to be true enough for practical purposes without being true in all cases. The legal challenges posed by chimerism extend beyond the dramatic cases that have made international news. They touch on fundamental questions about how the law defines identity, parenthood, and guilt. DNA evidence has been treated in courts as among the most reliable forms of forensic proof available, and with good reason, because for the vast majority of cases involving typical individuals with a single genome, a properly collected and analyzed DNA sample provides a highly accurate link between a biological trace and an individual. The problem with chimerism is not that DNA evidence is wrong. The problem is that it is incomplete in ways that standard protocols do not account for. A chimera who commits a crime might leave biological material at the scene that matches one of their two cell lines. If the forensic sample taken from them afterward happens to sample the other cell line, the match will fail. Conversely, if a chimera is innocent of a crime but happens to share one of their cell lines with the actual perpetrator, a genetic match could incorrectly implicate them, not through any malfunction of the science, but through a misunderstanding of what the science is actually measuring. Current forensic protocols typically sample a single tissue, usually blood or a cheek swab, and assume that the result represents the individual completely. For chimeras, this is not true, and the error may never be discovered unless the inconsistency is noticed and followed up with additional testing. In the Lydia Fairchild case and others like it, the inconsistency was noticed only because an obvious biological fact, a woman giving birth to a child, contradicted the genetic conclusion. In less dramatic cases, an incorrect genetic conclusion might simply stand unchallenged. The legal system has begun to develop awareness of chimerism, and some jurisdictions now have procedures for following up anomalous DNA results with multi-tissue testing. But chimerism has not been incorporated into standard forensic protocols in most places, and the possibility that a defendant might be a chimera is not routinely considered when DNA evidence is inconclusive or internally inconsistent. The deeper legal question is philosophical as well as practical. The law treats a person as a single legal entity with a single genetic identity, responsible for all actions performed by the body that identity inhabits. A chimera complicates this in ways that have no clear resolution. If one cell line in a chimera's body produced evidence found at a crime scene, but the person's legal identity is associated with the other cell line, what exactly has been proved? If a chimera has children genetically related to one of their cell lines, but not the other, which cell line defines the legal parent? These are not merely theoretical puzzles. They are live questions in actual legal cases, and the legal system is working out answers to them, case by case, with the help of forensic geneticists and medical experts, in a process that is still very much in progress. What chimerism ultimately forces the legal system to confront is that the concept of a unique individual, which underpins every aspect of the law from criminal liability to parenthood to inheritance, is a social and legal construct that rests on a biological assumption. That assumption is almost always correct, but almost always is not always. And the exceptions, rare as they are, expose the seam between the legal concept and the biological reality, demanding that one or both be revised to accommodate the truth. The most likely long-term resolution is not a fundamental rethinking of legal identity, but the development of more sophisticated forensic protocols that account for the possibility of chimerism when evidence is anomalous, combined with broader medical awareness that allows chimeric individuals to be identified and their condition properly documented. This would not require changing how we think about persons under the law, but simply improving the quality of the evidence we use to identify them. Not all human chimeras acquired their second genome before birth. Some become chimeras later in life, through medical interventions that introduce a second set of cells into a body that has already established its own genetic identity. The most common form of acquired chimerism is bone marrow transplantation, a treatment for certain blood cancers and other blood disorders in which the patient's own bone marrow is destroyed and replaced with donor marrow. The donor marrow contains hematopoietic stem cells, the cells responsible for producing all blood cells, and when these cells take up residence in the recipient's bone marrow and begin producing blood, the recipient becomes a chimera. Their blood cells are now being produced by donor cells carrying donor DNA, while the rest of their body, their skin, their muscles, their organs, continues to carry their original genome. A successfully transplanted patient will, for the rest of their life, have two distinct genomes in their body. A blood test will return the donor's genetic profile. A cheek swab will return their own original genetic profile. They are, in the biological sense, two people, and the fraction of their body that carries the donor genome can be measured and tracked over time, a process called chimerism monitoring that is now a standard part of post-transplant care. The monitoring matters because the degree of chimerism after transplant is medically significant. Full donor chimerism, where the recipient's original bone marrow cells are completely replaced by donor cells, is the goal in most transplant scenarios, because residual host cells may include the cancerous or dysfunctional cells that necessitated the transplant in the first place. Incomplete chimerism, where a mixture of host and donor cells persists, is called mixed chimerism, and it requires careful medical management. Blood transfusion is a less extreme version of acquired chimerism. When you receive someone else's blood, you receive their red blood cells, which carry their DNA, and those cells circulate in your body for the duration of their lifespan, typically about 120 days for red blood cells, and then die. This is transient chimerism, lasting only weeks to months, but during that period a blood test would technically detect DNA from both the recipient and the donor. Organ transplantation also produces chimerism. Any transplanted organ contains cells from the donor, and some of those cells, particularly immune cells called lymphocytes that are distributed throughout the organ, may survive in the recipient for years. The degree of this chimerism is generally small, but it is detectable with sensitive enough methods, and it has occasionally produced the same kind of anomalous DNA test results that more dramatic forms of chimerism create. The existence of transplant chimerism has an important practical implication that is not always appreciated. If a person who has received a bone marrow transplant later becomes involved in a criminal investigation, forensic DNA testing of their blood will return the donor's genetic profile rather than their own. This could, in principle, produce a false match or a false exclusion, depending on the circumstances, and forensic investigators are now advised to ask about transplant history when DNA test results are anomalous or unexpected. There is something philosophically interesting about transplant chimerism that distinguishes it from the congenital forms. In congenital chimerism, the second cell line was present from before birth or from very early childhood, integrated into the body at a developmental stage when the immune system was still learning to define self. In transplant chimerism, the second cell line arrived in an adult body with a fully mature immune system that had to be specifically suppressed to allow the foreign cells to take hold. The chimerism is intentional and therapeutic rather than incidental and developmental, and the person is fully aware that they carry donor cells in their body. This awareness does not change the biology, but it changes the experience. A transplant recipient knows they are a chimera. They know which tissues carry the donor genome and which carry their own. They live with this knowledge as part of their medical history, and the chimerism is monitored rather than accidentally discovered. In this sense, acquired chimerism is the most transparent form, the one in which the second genetic identity is not hidden but documented, tracked, and medically managed throughout the recipient's life. Everything discussed so far involves chimerism at a relatively large scale, where a significant fraction of the body's cells carry a second genome. But there is a subtler and far more widespread form of chimerism that operates at the level of individual cells scattered through tissues, present in such small numbers that standard genetic testing would never detect them, but detectable by sensitive modern molecular methods. This is called microchimerism, and it is, based on current evidence, essentially universal in humans. Microchimerism refers to the presence of a small number of cells from one individual within the body of another, where the transferred cells constitute a tiny fraction of the total, perhaps 1 in 1,000 or 1 in 10,000 or even fewer, but where those cells are genetically distinct from the host and can be identified by their different DNA sequence. The most common source of microchimerism in humans is pregnancy. During every pregnancy, a small number of cells cross the placental barrier in both directions. Fetal cells enter the maternal circulation and can travel to virtually any organ in the mother's body, where some of them take up residence and persist for years, sometimes for the rest of her life. At the same time, maternal cells enter the fetal circulation and can similarly distribute through the developing fetus's tissues. This two-way cellular traffic is a normal feature of human pregnancy, not a medical complication, and it leaves every person who has ever been pregnant and every person who has ever been a fetus with a small population of genetically foreign cells distributed through their body. The numbers are small. In a typical woman who has had children, fetal microchimerism might affect 1 in 10,000 cells in a given tissue, or 1 in a million, or even fewer, depending on the tissue and the sensitivity of the measurement. These cells would not be detected by standard DNA testing, which measures the average genetic signal of many cells rather than identifying individual variant cells. They can be detected by highly sensitive molecular methods that amplify rare sequences, methods developed in research settings over the past 2 to 3 decades. What these methods have revealed is that microchimerism is not an occasional curiosity but a near universal feature of human biology, that the notion of a body containing exactly one genome is a simplification that does not fully describe how human bodies actually work, and that the cellular boundaries between individuals are more permeable than standard biology textbooks have traditionally suggested. The implications of microchimerism for health, for disease, for the immune system, for the biology of pregnancy itself, are areas of active research. And what the research has found is surprising, and in some cases profoundly counterintuitive. Perhaps the most counterintuitive finding is that the immune system does not simply destroy these foreign cells when it encounters them. A healthy immune system is extraordinarily good at identifying and eliminating non-self cells, which is why organ transplants require powerful immunosuppression to prevent rejection. And yet microchimeric cells, present in much smaller numbers than a transplanted organ, but equally foreign in genetic terms, persist for decades without being eliminated. They have learned to evade or co-opt the immune surveillance that would normally remove them, and the mechanisms by which they do this are still being investigated. One leading hypothesis is that tolerance to microchimeric cells is established during fetal development, when the immune system is still maturing and learning which cells to leave alone. If a fetus is exposed to maternal cells during a period when the immune system is calibrating its definition of self, those maternal cells may be incorporated into that definition, effectively being treated as self for the rest of the individual's life. The same process may work in the other direction, with fetal cells establishing tolerance in the maternal immune system through mechanisms that are currently poorly understood but are the subject of intensive research. What is clear is that the relationship between the immune system and microchimeric cells is not simply one of tolerance. The microchimeric cells are active participants in the host's biology, interacting with the immune system in ways that appear to have both beneficial and potentially harmful consequences, depending on the circumstances and the tissue where those cells reside. The discovery that fetal cells persist in the mother's body for decades after pregnancy was first made in the early 1990s, and it has since been confirmed and extended by dozens of studies across multiple research groups. The persistence is remarkable. Fetal cells have been found in the blood, the liver, the lungs, the kidneys, the thyroid, the heart, and the brain of women who gave birth decades earlier. Studies of women who died in their 70s and 80s have found fetal cells in their brains, cells with the genetic signature of their son's Y chromosomes, cells that had been living quietly in the brain tissue for 40 or 50 years after the pregnancy ended. These are not dead cells preserved in tissue. They are living cells, metabolically active, integrated into the tissue structure of the organs that harbor them. They behave, in many cases, like stem cells, capable of differentiating into the cell types appropriate for the tissue where they reside. Fetal cells found in the liver of a woman who had a pregnancy decades earlier may have become liver cells, functioning as liver cells, indistinguishable in their behavior from the woman's own liver cells, while carrying the fetal genome in their nucleus. This raises a remarkable possibility. The fetal cells that entered the mother's body during pregnancy may not have been simply passing through. They may have integrated into the mother's tissues and contributed, in some small way, to the function of those tissues for the rest of her life. A mother's heart may contain a few dozen cells carrying her child's DNA, quietly beating alongside her own cardiac cells, incorporated so completely that they require sophisticated molecular methods to detect. The reverse is also true. Maternal cells enter the fetal circulation during pregnancy, and some of those cells persist in the child after birth. A study published in 2009 found maternal cells in the brains of fetuses that had been examined after loss at various stages of pregnancy, indicating that maternal cells cross the placental barrier early and distribute widely, and research has confirmed that maternal microchimerism persists in children into adulthood, with maternal cells found in the blood and tissues of grown adults many decades after their birth. This means that virtually every person carries some cells from their mother, and that virtually every woman who has been pregnant carries some cells from each of her children, and that in families with multiple pregnancies, siblings may have exchanged cells through the maternal body as an intermediary, since a pregnant woman carrying cells from of those cells to a subsequent fetus, so that a younger sibling might carry cells that originated with an older sibling, passed through the mother's body, and transferred to the younger child. During genomes, the human body, viewed through the lens of microchimerism, is not a sealed genetic fortress, but a porous biological entity that has exchanged cells with every person it has been in close enough contact with during vulnerable developmental windows, and it carries those exchanged cells for the rest of its life. The full map of microchimerism in any individual would be extraordinarily complex to reconstruct. A woman who has had 3 children would carry cells from each of them, cells from her own mother, and potentially cells from older siblings passed through her mother during gestation. Each of her children would carry cells from her, and possibly cells from each other as transferred through her body during subsequent pregnancies. The cellular connections that link family members across generations are not merely genetic, inherited through the copying of DNA from parent to child, but also literally physical. Small populations of living cells that have crossed the boundaries between individuals and persisted, carrying their original genome in bodies they were not born into. The biological consequences of having foreign cells distributed through your body, even in small numbers, are complex and not yet fully understood. Research over the past 3 decades has produced evidence pointing in different directions, suggesting that microchimerism can be both beneficial and harmful, depending on circumstances that are still being worked out. The immune question is the central puzzle. The immune system is designed to distinguish self from non-self, to attack cells carrying foreign genetic signatures while leaving the body's own cells alone. A chimera should present a constant immune challenge, because the second cell line carries a foreign genome that the immune system would normally recognize and destroy. Yet chimeras exist, often without any sign of immune conflict, which means the immune system has somehow learned to tolerate the second cell line as if it were self. How this tolerance develops is an area of active research. One hypothesis is that during fetal development, when the immune system is still learning which cells to tolerate and which to attack, the presence of foreign cells from very early in development teaches the developing immune system to accept those cells as self. If chimerism is established before the immune system has fully matured, the second cell line may simply be incorporated into the definition of self from the beginning, sparing it from the immune attack it would face if introduced later in life. The consequences of this tolerance for health outcomes are significant. On one hand, the immune tolerance of the chimeric cell line may be beneficial, helping to reduce inflammation and damping autoimmune responses. On the other hand, the same tolerance that spares the second cell line from attack may also make it harder for the immune system to clear any cells of that line that become abnormal or malignant. Studies of microchimerism and disease have produced intriguing and sometimes contradictory findings. Some research has found associations between high levels of fetal microchimerism in mothers and lower rates of certain cancers, particularly breast cancer, suggesting that fetal cells may play some protective role. Other research has found associations between microchimerism and autoimmune diseases, including thyroid disease, scleroderma, and lupus, where the presence of genetically foreign cells in the body may contribute to the chronic immune activation that characterizes these conditions. The most carefully studied association is between fetal microchimerism and thyroid disease in women. The thyroid is a tissue where fetal cells have been found in relatively high concentrations, and several studies have found that women with autoimmune thyroid diseases have higher levels of fetal microchimerism in their thyroid tissue than healthy controls. Whether the fetal cells are causing the autoimmunity, responding to it, or are present for entirely unrelated reasons remains an open question. But the correlation is consistent across multiple studies conducted in different countries. The biological role of fetal cells in the maternal brain is perhaps the most tantalizing open question in the field. The brain is an immunologically privileged site, meaning it is partly protected from the standard immune surveillance that operates elsewhere in the body, and fetal cells that reach the brain may persist there in larger numbers than in other tissues precisely because the brain's immune environment is more permissive. What those cells are doing in the maternal brain, whether they are integrated into neural circuits, whether they influence behavior or cognition or mood, whether they contribute to the well-documented biological changes that occur in mothers following pregnancy, is genuinely unknown, and answering these questions will require decades of additional research. The study of chimerism and disease is still in its early stages, and many of the associations reported in the literature are based on relatively small studies that have not yet been replicated at scale. The field is growing rapidly, however, and the development of more sensitive detection methods and larger biobanks of human tissue samples is enabling studies that were not possible a decade ago. In the coming years, the picture of how microchimeric cells affect health will become considerably clearer, and the answers may fundamentally change how we understand the biology of pregnancy, autoimmunity, and the long-term relationship between generations. Among all the questions that chimerism raises, the most unsettling is the one it poses about personal identity. We treat identity as singular. You are one person. You have one genome, one self, one continuous experience that began when you were born and will end when you die. Your cells are yours, and together they constitute the biological substrate of the you that thinks and feels and moves through the world. Chimerism challenges this picture not by proving it wrong, but by revealing it as a simplification, a useful and usually accurate approximation that happens to be incomplete in ways that matter under certain circumstances. A tetragametic chimera, one who developed from the fusion of two zygotes in early development, is in a profound biological sense two people. Not in the sense of having two personalities or two streams of consciousness, because the brain, which is the substrate of experience, is a single integrated organ, and the experience of being a chimera is identical to the experience of being a non-chimera, which is to say that there is no experience of being a chimera from the inside. There is simply the experience of being a person, living and thinking and moving through the world, unaware that the cells doing the living are not all running the same genetic program. The philosophical question is whether this biological duality matters for the concept of self. Most philosophers who have addressed the question conclude that it does not, because selfhood is constituted by psychological continuity, by the continuous stream of memories and experiences and personality, and not by genetic identity. A chimera is no more two people in the philosophically meaningful sense than a person who has received a bone marrow transplant is two people, because the transplanted cells do not bring a new consciousness or a new set of memories or a new personality. They are simply doing the work of making blood in a body that continues to have one continuous experience of being a self. But the biological duality does matter in a different sense, because it reveals that the boundary between one person and another at the cellular level is not where we assumed it was. The genome has been treated, particularly in legal and forensic contexts, as the definitive marker of individual identity, the one thing that is uniquely yours, that proves who you are and connects you to your actions and your relationships. Chimerism reveals that this identity marker is not always singular, and that a body can contain more than one genetic identity simultaneously. The discovery of chimerism is, in a small way, the discovery that human individuality is more complex than our social and legal categories assume. We are built to think about persons as discrete, bounded, genetically unique individuals, and almost always, this is the right way to think. But the edges of that category, examined closely enough, turn out to be softer than they appear, the boundaries between one person and another more permeable, the concept of a single unique biological self more complicated than the simple story we tell about it. The experience of learning you are a chimera, for those who have learned it, is reported as disorienting but not shattering. The people who discovered their chimerism in the course of medical investigations or legal disputes consistently describe the revelation as strange and intellectually destabilizing, but not as something that changes who they feel themselves to be. The self is more robust than the genome. Identity is written in experience, not just in DNA. The chimera is still the same person they were before the test, still living the same life, still recognizing the same face in the mirror every morning. And the second genome in their cells does not speak to them, does not offer a competing perspective, does not divide the experience of being alive into two streams. It simply is there, in the cells, doing its quiet work. One of the most frequently asked questions about chimerism is simply, how common is it? The honest answer is that we do not know with precision, and the uncertainty is itself informative. For full chimerism of the kind that produces two clearly distinct cell lines throughout the body, the kind detected in the blood type cases and the maternity cases and the criminal cases, estimates vary widely. Some researchers have suggested that 1 in 100 people may be chimeras of this kind, while others have estimated the figure at 1 in 1,000 or 1 in 10,000. And the truth is that without population-level screening using methods specifically designed to detect chimerism, we simply cannot be confident about the prevalence. The difficulty is that chimerism is essentially invisible unless you are specifically looking for it. A routine blood test that returns a single blood type does not prove the person is not a chimera, because a chimera, whose bone marrow is dominated by one cell line, will have a single blood type, and the second cell line might be concentrated in the liver, or the skin, or some other tissue not routinely sampled. To reliably detect chimerism, you need to sample multiple tissues and compare the results. And this is not something that is done in standard medical practice. The cases that have been documented and confirmed are almost certainly a small fraction of all chimeras who exist, because they are the cases where chimerism surfaced because of an anomalous test result that prompted investigation. A chimera who never has a blood type test, never undergoes transplant compatibility testing, never has a DNA maternity or paternity test, and never becomes involved in a forensic investigation, will in all probability never know they are a chimera. For microchimerism, the situation is clearer and the prevalence is much higher. Studies using sensitive molecular methods to detect fetal microchimerism in women who have had pregnancies find evidence of it in the great majority of women tested, and the persistence of those cells for decades has been confirmed repeatedly. Maternal microchimerism in children appears to be similarly prevalent. If microchimerism is included in the definition, then chimerism, broadly construed, is not a rare curiosity, but a near-universal feature of human biology. The clinical and social implications of widespread undetected chimerism are not fully appreciated. How many legal decisions about parenthood rest on DNA evidence obtained from a chimeric individual who happened to be sampled at the wrong tissue? How many forensic conclusions have been drawn from genetic evidence that accurately reflects one part of a chimeric person's body, but not the whole? How many individuals are carrying a second genome through their lives without knowing it? A genome that could, under some circumstances, produce a legal or medical anomaly that they would be completely unprepared to explain. These are not rhetorical questions. They are genuine gaps in our current understanding, and filling them will require both better detection methods and broader awareness among the clinicians, lawyers, and investigators who work with genetic evidence. There is also a deeper question embedded in the prevalence problem, which is whether chimerism should be understood as a variation or an aberration. For most of medical history, it has been treated as a curiosity, a rare anomaly worth documenting, but not worth systematically screening for. The accumulation of evidence for widespread microchimerism challenges this framing. If virtually every person who has ever been pregnant carries cells from their children, and virtually every person carries cells from their mother, then what we have been calling chimerism in its subtler forms is not an aberration at all, but a standard feature of human biology, one that happened to go undetected for most of history simply because we lacked the tools to look for it. The methods used to detect chimerism have improved dramatically over the past 3 decades, and the improvement has both revealed more chimeras and raised the question of what to do with that information once it is found. The earliest detections of chimerism relied on obvious anomalies: a person with 2 blood types, a DNA test that returned inconsistent results across samples. These anomalies are dramatic enough to prompt investigation, but they represent only the most extreme and detectable end of a spectrum. The development of more sensitive molecular methods has allowed researchers to detect chimerism at much lower levels, finding a few chimeric cells in a tissue where thousands of host cells surround them. The most powerful current methods use what is called short tandem repeat analysis, which examines specific regions of the genome where the number of repeated sequences varies between individuals. By identifying the STR profile of a known cell line, such as a bone marrow donor, and then searching for that profile in samples from the recipient, researchers can detect chimerism at levels below 1% of the cell population. Even more sensitive methods using next-generation sequencing can detect foreign cell populations at even lower levels, approaching 1 in 10,000 cells in favorable circumstances. For clinical chimerism monitoring after bone marrow transplant, these methods are now standard of care, because tracking the ratio of donor to host cells over time is essential for managing transplant outcomes. For research into natural chimerism and microchimerism, the same methods have been adapted to look for naturally occurring genetic differences between host cells and the small population of foreign cells they harbor. One challenge specific to natural chimerism, as opposed to transplant chimerism, is that you often do not know in advance what genome to look for. In transplant chimerism, you have the donor's genetic profile to use as a reference. In natural chimerism arising from a vanished twin or early embryo fusion, you may not have any independent sample of the second genome. You have to detect it by its difference from the host, which requires either an obvious discordance in a known genetic marker or a comprehensive analysis of the genome, looking for cells that do not fit the predominant pattern. The development of methods that can sequence the genome of individual cells, called single-cell genomics, has opened new possibilities for detecting chimerism in tissues where it might be present at very low levels. These methods are currently expensive and technically demanding, but as with all sequencing technologies, they are becoming faster and cheaper rapidly, and within the next decade they may become practical tools for routine clinical screening. What will happen when chimerism screening becomes practical and affordable enough for widespread use is a question with no clear answer. The discovery that one is a chimera, carrying a second genome derived from a twin who never lived, is not medically dangerous in most cases, but it is psychologically significant, and the legal implications, as the cases already documented have shown, can be far-reaching. Society will need to develop frameworks for understanding and responding to this information, frameworks that neither overreact to the biological complexity nor ignore its real practical consequences. The genetics community has also begun to grapple with the question of what to do when chimerism is discovered incidentally, as a byproduct of genetic testing performed for another purpose. As whole genome sequencing becomes cheaper and more widely used in clinical medicine, incidental discoveries of chimerism will become more common, and clinicians will need guidance on how to counsel patients who receive this information. The discovery that one carries a second genome, particularly if that genome is identified as originating from a vanished twin, raises questions about identity and biology that standard genetic counseling frameworks were not designed to address. As you lie in the dark tonight, consider the cells that make you. There are approximately 37 trillion of them, and each one carries, in its nucleus, the instructions for building a human being. Most of those cells are running your instructions, expressing your genes, doing the work of keeping you alive according to the program written in your DNA at the moment of your conception. But some of them, perhaps, are running a different program. Perhaps the program of a twin who shared the womb with you for a few days or a few weeks before being absorbed into the body that became you. Perhaps the program of your mother, who sent cells across the placenta during the months she carried you. And some of those cells took up residence in your tissues and are still there now, quietly doing their part. Perhaps, if you have had children, the program of a child who left a few of their cells behind when they were born, cells that found their way to your liver or your heart or the quiet interior of your brain and have been living there ever since. The body you inhabit is, in this sense, not entirely yours, and it may never have been. This is not a disturbing conclusion, or at least it need not be. The self that thinks and feels and experiences is not located in any individual cell or any particular genome, but in the integrated activity of the brain, in the continuity of memory and experience, in the patterns of personality and perception that have developed over a lifetime. A few genetically foreign cells in the liver do not divide the self. They do not create two inner voices or two competing experiences. They are simply cells, doing cellular work, integrated into a body that experiences itself as one. But the discovery of chimerism does something important to our understanding of what biological individuality means. It reveals that the sharp boundary we draw between one person and another, the boundary that legal systems and forensic science and our intuitions about identity all assume exists, is not as sharp as we thought. Cells cross that boundary during pregnancy. They cross it during transplantation. They may have crossed it during embryonic development, in the first days of life when the rules about who will become whom had not yet been written. The science of chimerism is also a story about how scientific knowledge advances by finding exceptions to rules we thought were absolute. The rule was, one person, one genome. The exception is, not always. The exception, once found, does not invalidate the rule for the vast majority of cases, but it demands that the rule be understood as an approximation, useful and usually accurate, but incomplete in ways that matter when you look closely enough. That kind of revision, from simple rule to complex approximation, happens again and again in the history of science, and it is always an improvement, because the world is not simple, and the models that match it most closely are the ones that are willing to accommodate its complexity. Chimerism is the world being more interesting than our models assumed, and the scientists who noticed the anomalies and pursued them rather than dismissing them gave us all a richer, more accurate, more wondrous picture of what a human body actually is. There was, perhaps, another person who shared your beginning. They did not survive, but something of them may have, encoded in the cells of the body that became yours, present without your knowledge for every day of your life, as close to you as your own heartbeat. You carry them, and you always have, and in the cellular machinery of your body, something of that early companionship persists, quiet and invisible and intimate beyond any other closeness, two genomes learning, from the very beginning, to share a life. Sleep well. You are not as alone as you thought, and that is, in the end, a beautiful thing.